Thanks, Dan, and thanks to the organizers. It's terrific to be here. I'm learning buckets, and will continue to. So most of the talk will be establishing a proof of principle to show how far you can get in the analysis, and in the mechanistic analysis of a mammalian social behavior. And it turns out that if you choose your problem simple enough, that you can demonstrate specific genes expressed in specific neurons essential for a specific mammalian social behavior. So here's the problem. This is a behavior that you're probably familiar with called lordosis behavior. It's expressed by female four-footed animals, but not by male four-footed animals. It's controlled exclusively by estrogens and, following estrogens, progesterone. And it controls reproduction by quadruped mammals, because without this behavior, fertilization cannot occur. Put simply, we understand mechanisms from receptor chemistry measured in angstroms through seasonality measured in light years. And having discovered the relevant hormone receptors for these behaviors and worked out the neural circuit for it, all of this is published, and so I'll go quickly. The genes regulating lordosis behavior discovered so far have two properties. First, that after estrogens bind to estrogen receptor alpha, or ER beta, these transcripts are elevated, and following binding and translation of these proteins facilitate female reproductive behaviors. Of special interest for this talk will be the progesterone receptor gene. And to prove the necessity of estrogens binding to estrogen receptor alpha, this gene product, in ventromedial hypothalamic neurons, we use an AAV viral vector microinjected specifically into the ventromedial nucleus of the hypothalamus. And this viral vector codes an siRNA against estrogen receptor alpha compared to the control vector. And you can see that following the siRNA against estrogen receptor alpha, proceptive behaviors go to 0, lordosis behaviors go to 0. Instead, the females reject the male, usually by kicking him in the ribs. And so the question is how this transcription factor does its job. And to explain that briefly, we have to have a brief exercise in chromatin chemistry. And this was mainly discovered in yeast, and it's been elucidated most brilliantly by one of our colleagues at Rockefeller, namely David Alice. And the easiest way to start is to say that in the cell nucleus, there's no such thing as naked DNA. Instead, you have inactive DNA, which is wound around histones, transformed into DNA that's open enough for transcription factors and coactivators to land and do their job. And the easiest way to see how this happens is to take a side view on these nucleosomes. And again discovered mainly in yeast, a process initiated by a basic protein called histone H1. You have a bunch of histones coalesced with tight protein-protein interactions around which is wrapped inactive DNA. And the saving grace is that the histone tails sticking out, the N termini of these tails, have lysines and arginines, which are subject to various chemical modifications and manipulations. For example, I'll use the acetylation, which is pro-transcription, and it facilitates transcription, perhaps by busting up protein-protein interactions in the nucleosome. And as a result of that, you have open DNA, and here we have estrogen receptors binding as dimers to estrogen response elements, glucocorticoid receptors, as both Michaels talked about, and also Bruce, bound to its cognate glucocorticoid response element, in both cases controlling transcription of their specific hormone receptive genes. So to bring this back to the problem of discovering how estrogens act in ventromedial hypothalamic nerve cells to cause lordosis behavior, what we do first is immunocytochemistry, where we use antibodies in this panel against estrogen receptor alpha. Here are the green cells, all in the ventromedial nucleus of the hypothalamus, the cells that are responsible for governing lordosis behavior. Here, the antibody specific to pan-acetyl H4, and here's the merge. So all of the yellow cells are ER-alpha expressing cells which have this acetylated H4, histone 4, and as a function of estrogen treatment, time after estrogen treatment, you have a significant increase in the number of the yellow cells. You can collect data in the relevant cell group, that is the ventromedial hypothalamus, a lot faster using Western blots. And this was a pleasing slide in the sense that two transcription facilitating marks, namely pan-acetyl histone 3 and an antibody that recognizes a combination event of an acetylation event and a phosphorylation event in these ventromedial hypothalamic cells, increase as a function of time after estrogen treatment, whereas a repressive mark decreases. This is all far previous to the time that lordosis will occur, which is 18 hours after estrogen treatment. And we want to apply this kind of thinking, which is across the genome in these particular neurons, to the transcription of the progesterone receptor, which I mentioned, which is not only estrogen sensitive, but it's also tightly linked with the evocation of lordosis behavior. So here, the same postdoctoral fellow, Katuna Gagnidze, did an immunocytochemical study. There's actually two transcripts coming from the progesterone receptor gene, the large one, the big one, and the smaller one, and both of them are higher level of immunocytochemical product 6 hours after estrogen treatment, especially progesterone receptor B. So what we want to do then is make use of our knowledge of specific genes. We don't want to be limited to genome-wide histone facilitation, or global histone facilitation. So the chromatin immunoprecipitation method yields promoter specificity, even though you don't get neuron by neuron specificity out of the same data. So what you do is you take chromatin from your brain region of interest, in the case of this talk, ventromedial hypothalamus, for analyzing this lordosis behavior. You sonicate it. Your first cut is immunoprecipitation, where you use the antibody, which is specific to a particular histone moiety, in this case an acetylated histone tail, and at that you use that as the starting point to subject to a progesterone receptor specific primer, and then subject that to PCR, thus to look at acetylation specifically on the progesterone receptor promoter. And the results are that 3 or 5 hours after estrogen treatment in the ventromedial nucleus of the hypothalamus, you get this specific histone modification on the progesterone receptor promoter, but it's complicated. It's a function of time, it's a function of the histone mark studied, and the promoter segment. So here are the promoter segments that we studied. Here's the transcription start site. Here's the encoding region for PRB, PRA. We're studying both acetyl, H3 acetyl, and also a trimethylation, which in this case is a transcription facilitating. And you can see that on this particular promoter segment, we get an estrogen effect, in this case 3 hours, and in this case 5 hours after estrogen treatment. So you have to look at each promoter segment as a function of time. Now, up to this point, all of the things have been correlational. The same estrogen treatment and the same neurons that leads to lordosis behavior, if you put the estrogen next to those neurons, also leads to the histone changes. What about an active manipulation in which you can cause a lordosis change? In this case, Zachary Weil, before he took a position at Ohio State, used a histone deacetylase inhibitor microinjected specifically into the ventromedial hypothalamus, and we set up the animals so that they didn't have enough estrogen so that they would do this female reproductive behavior by themselves without any further treatment. And then when he gives the histone deacetylase inhibitor, that is, there's more acetylation in the ventromedial hypothalamus, he gets lordosis behavior and a decreased rate of rejection. How do things get to be this way? As I said at the beginning, this particular social behavior, essential for reproduction, is shown by females but not by males in a wide variety of mammalian species. And so what we want to do is to look at sex difference in gene transcription in the hypothalamus, or perhaps the preoptic area, which could account for the behavioral difference, and following the discovery of gene transcription, to look at differences in the protein chemistry of histone tails in the same neurons. So many of you know that in laboratory animals like rats and mice, a single injection of testosterone on the day of birth will turn these animals phenotypically from behavioral females into behavioral males. Or to be more specific, female typical reproductive behaviors under the controls of estrogen and progesterone go to 0. And by the way, the main neuroendocrine characteristic in these animals that's female specific, the ovulatory release of luteinizing hormone, also goes to 0. And so that turns the question that I asked a couple of slides ago into the following question: how does a single testosterone exposure, which will go away after all, lead to a lifelong change in behavior? And once again, we want to look at sex differences in transcription, followed by, in separate experiments, differences in protein chemistry of histone tails. So the microarray experiment was organized this way. Here's a rough sketch of the central nervous system sensitivity to testosterone in animals like rats and mice. And what we wanted to do was to take our hypothalamic and preoptic tissue for a microarray late enough that the early genomic actions of testosterone, that is this part of the curve, would have taken place, and the brain actually would be masculinized with respect to these behaviors. And secondly, we wanted it to be early enough that the transcriptional changes that we see are causal to all subsequent behaviors rather than as a result of sexually differentiated behaviors that had already developed. And so this timing turned out to be optimal. And what we have here, just looking at it briefly, is in the preoptic area tissue, which is essential for male sex behavior, and the medial basal hypothalamic tissue, let's call it ventromedial nucleus of the hypothalamus, essential for female sex behavior. Males on the y-axis, females on the abscissa, over 11,000 genes. And in the preoptic area, amazingly, since these are log-log coordinates, by the way, in the preoptic area, an amazing 8% of transcripts are significantly different between females and males. And in the medial basal hypothalamus, 0.9%, which still calculates out to be about 110 genes. For purposes of time, I'm not going to talk about individual genes except briefly about this one. And I'm happy to send the initial list of genes to anybody in the audience who emails me. To be mercifully brief, what I've done is to summarize categories of genes. In this case, significantly greater in neonatal male preoptic area or hypothalamus. It's going to turn out to be that there's a total of 94 such categories. And you can see, I'm not going to read these, all you can do is scan your eyes down the list and see the amazing range of biochemical functions that are represented. And then on the next slide, again to be mercifully brief, just categories of genes. In this case, significantly greater in neonatal female preoptic area hypothalamus than males, total of 94 between the two slides. And I might point out that even though I'm doing this in the service of explanation of a simple reproductive behavior, it could be relevant for many different behaviors which have the dual properties of being subject to early stress and having a sexually differentiated phenotype. I said I'd follow up with just one gene, connexin. Connexin is an especially interesting gene because barrels of 6 connexin 36 proteins make the gap junctions, which form these tiny holes between adjacent neurons and allow for the transfer of electrically charged particles from one nerve cell to the other, thus providing a rapid alternative to synaptic transmission. And it's both predicted and unpredicted. On the one hand, we do, I'm off my, talking about a different slide. Here we see sex differences, males greater than females, both in the ventromedial hypothalamus and in the preoptic area, differences using qPCR for messenger RNA levels in the VMN and POA. And as is true with many genes of developmental importance, it's an evanescent rise, or in this case, an evanescent sex difference, because by day 2 already, after the period of sexual differentiation of the brain, the significant difference has disappeared here and has been reduced here. And now the question is, how do long-lasting changes of gene expression, as might be important for sexually differentiated reproductive and, for that matter, maternal behaviors, come about? And what we want to do is to be theory-driven and to say, what is the simplest possible theory with respect to the medial preoptic neurons, which are responsible for male sex behavior, and the ventromedial hypothalamic neurons, which are responsible for female sex behavior? What's the simplest possible theory with respect to histone tail chemical changes? And so in the genetic male, the simplest thing to say is that you'd be looking for a lot of acetylation marks in preoptic neurons, thus to facilitate facilitate transcription in the neurons that are responsible for male sex behavior, and you'd be looking for repression, defeminized behavior, in the ventromedial hypothalamus, whereas in the genetic female, you'd want exactly the opposite. Methylation among a lot of genes in the preoptic area, and acetylation in the ventromedial hypothalamus. And it turns out that we do indeed have sex differences, but the results don't fit the theory at all. Here we have a significant sex difference, males greater than females, here too. But if the results fit the theory, in the preoptic area responsible for male sex behavior, this red bar would be a lot higher than this blue bar, and likewise in the ventromedial hypothalamus, these red bars would be a lot higher than the blue bars. Well, you could say you're looking across all the chromatin there is in the genome, and that's probably the source of your problems. Therefore, you should, theoretical problems. Therefore, you should move to CHIP right away, and indeed we will and are. Here's the raw data over here, by the way. But I must say that even using this global histone technique, we are able to demonstrate the influence of neonatal testosterone, which masculinizes the brain, and an endocrine disruptor, ethinyl estradiol. So here in the ventromedial hypothalamus and here in the preoptic area, the blue bars, the males, have significantly greater amounts of this pro-transcription mark, histone 3, lysine 4 trimethylation, than the females. And when we masculinize the brain by giving the females testosterone propionate, this bar right here, we bring the results back toward the male level. Here also the same pattern, not statistically significant. And now in this experiment, on the bottom half of the slide, we're using the endocrine disruptor ethinyl estradiol. It's a little bit like the BPA that Francis was talking about. And here we have, again, the males significantly greater than the females with respect to this lysine 4 trimethylation mark, with a significant move in the endocrine disrupted females back toward the male level. And here also, males greater than females, and a significant move back toward the male level. But even though we came away from these two experiments with a little bit of data, still we have to move to the CHIP technique, which I illustrated. So having spent a lot of time going for what I would call deep conclusions with respect to the mechanistic analysis of normal behaviors, in especially sex differences in normal behaviors in simple laboratory animals, I'd like to finish up this talk by asking the rhetorical question, when do we really care about sex differentiation of the brain, especially with respect to the treatment of the two genders in modern societies? And the answer to that, I think, is when we consider sexually differentiated maladies. So in the States, the incidence of depression is reported to be about 2.7 times in female compared to male, chronic fatigue about 8 to 1, anorexia, bulimia primarily female, whereas ADHD more often male, acting out, externalizing problems, violence more frequent in males. But the syndrome that, the set of disorders that I've become especially fascinated with is autism. And autism, if you make a rough average, is present or diagnosed about 4 times as frequently in males as in females. And what I had to do was to analyze 3 literatures, to review 3 literatures, one about autism, one about early stress, and what about androgenic hormones, in order to come away with what I'll call a 3-hit theory. So by analogy to the 2-hit theory of cancer, which I think you know what that is, the 3-hit theory would be that in the neonatal prenatal neonatal male, you have the effects of early stresses piled on top of the effects of androgenic hormones, piled in turn on top of large numbers of genetic predispositions. And so, as a guy who's not a geneticist, I would say more than 40 genes have small effects, significant effects, on the appearance of autism. An expert like Joe Buxbaum at Mount Sinai would say more than 100. And so if now for the end of this talk, if we finish up with a couple of slides, let's take for granted these genetic predispositions. They're going to be there, small effects, and there may be, of course, more than one in any individual. And in the next slide, I'm going to do the easy part, which is to talk about androgenic hormone effects. And then in the last slide, the harder part about how these two things interact. So the androgenic hormone effects would be something like this, that in the neonatal brain, and I'm thinking about humans now at the end of the first trimester, if and only if testosterone has docked to the androgen receptor, it gets into the nucleus and binds as a dimer to the androgen response element. And I might point out that the concentrations and the locations of these proteins called coactivators, which are often histone-active enzymes, can be essential to the effectiveness of AR and could be responsible for large individual differences in the impact of testosterone on this gene expression or any gene expression and subsequent behaviors that depend upon those genes. So if we take the androgens as a relatively easy problem supported by an entire generation of molecular endocrinology, what does that mean for autism according to this three-hit theory? Remember, the first hit, we have the genetic predispositions in place. And now what I'm talking about as a result of a large literature is brainstem androgen-sensitive neurons producing arousal transmitters, which these neurons project up, among other places, into the amygdala. And it was on this very campus, UCal Irvine, where Benno Roosendahl and Jim McGaugh discovered that the deposition of norepinephrine into the amygdala makes for a supersensitive amygdala and enhanced fear behavior. I'm hypothesizing that in the presence of neonatal stresses, an adverse intrauterine environment, if you will, which in baby males is by definition social, that this supersensitive amygdala will generate a proclivity towards social anxiety and consequent to social anxiety, social avoidance, which I think of as a hallmark of autism, and I have a feeling I'm going to be corrected in a few minutes by Sir Michael. So what I'm thinking is that the combination of the genetic predisposition, the androgenic effects making for a supersensitive forebrain, and then a rough prenatal experience and or neonatal experience accounts for the male prevalence in autism. This is a testable theory. Within a month, we'll have catnap2 mutant mice in the lab. That's one of the genes that's prominently associated with autism, and we'll subject them to the obvious experiments that you would predict from this theory. So preparing for the meeting, I went through some of the papers of our conferees. Michael Rudder, more than 3 decades ago, with respect to the genetic factor of the three-hit theory, pointed out a concordance of diagnosis for autism, 36% in monozygotic twins, 0% in dizygotic twins, and with great prescience, in 1977, he talked about gene-environment interactions. Charles Nelson and his colleagues in 2009 reported a large study of Romanian preschool students who had been subjected to institutionalized environments, and especially in Table 4, they talked about gender-environmental interactions in which the males were more hurt by the impoverished upbringing than the females were. And by comparison to Frances' introduction this morning, where she has powerfully implicated DNA methylation in epigenetic effects, in this particular theory, I'm emphasizing histones, but that's partly just because I learned a lot of histone chemistry. Caveats, methodologic issues. Megan Gunner, in her 2009 paper, has warned us that results in these behavioral studies can vary according to stress paradigm. And so we have to be sensitive to that. And finally, Tom Boyce and his colleagues have shown in a rather large study a limited stability of autonomic status during development. And I was disappointed by that because I was depending upon the kinds of findings that you would remember from Jerry Kagan and from people like Mary Rothbard, who talked about considerable temperamental stability, which I need in my theory to account for persevering behavioral tendencies. So in summary, we understand mechanisms for simple biologically adaptive sexual differentiated behaviors in great detail. Hormone receptors following hormone binding, gene transcription, and as a result of that, the control of a neural circuitry which I've defined. And this hopefully provides a paradigm, at least a scientific paradigm, for understanding other emotionally laden behaviors. I would insist that methods for measuring histone N-terminus chemical modifications in order to explain long-lasting effects of early stresses on subsequent behaviors now can be applied broadly across the neuroscientific and behavioral realm. And we sincerely hope that this kind of work with laboratory animals will aid in approaching important subjects like the stress-sensitive aspects of autism, in my case, in the sexual differentiation. Thank you very much.